in which we use both experimental work and mathematical modeling to try and unravel some of the mechanisms involved in the movement of active particles and their collective behavior. And cell motility, that is the incessant swimming motion of particles, be them active, synthetic particles, or indeed real microswimmers, bacteria, phytoplankton, really exemplifies what we think of active matter. But even more than that, actually, these organisms are often subjected to complex, dynamic, external fields as well. And so we can see suspensions of microorganisms like this one here, subjected to shear flows. These could be in groundwater or in the ocean, and their rheology, their bulk properties might be very important. We can see organisms that actually follow magnetic field lines and can navigate along the magnetic field associated with the Earth to find low oxygen environments to grow and divide. And there are also a huge range of organisms that can detect and respond to chemical gradients in their microenvironment and swim towards food sources. So in addition to the kind of non-equilibrium nature of motility itself, we see a whole range of circumstances where the actual environment itself is also heavily out of equilibrium. And this has really interesting interplay with the motility and the movement of the organisms. So today I'm going to tell you a bit about these three stories that we've been working on. Now, it goes back a long time. We look at the work from Einstein to understand the rheology of a dilute suspension of passive particles and the leading order correction to the viscosity due to small particles suspended in that. And so this is a well-known correction up to the volume fraction phi. But something really peculiar and interesting happens really when you consider a suspension of microswimmers, even in the dilute limit, that actually whether or not the viscosity is enhanced or diminished with respect to the solvent fluid depends on whether they push themselves through the fluid with a flagellum on the back or whether they pull themselves through the fluid with flagella on the front. And so whether they're pushers or pullers can actually change the overall viscosity of the suspension, and this is well understood in the dilute limit based on how the flow field around an individual interacts with the external flow as you try and shear this fluid. But things get a little bit more complicated when we're interested in a dense suspension, that is an array of microorganisms for which we can't take a superposition of individual flow fields, but they start to interact with each other hydrodynamically. So to investigate this problem, we've used a canonical model framework in low Reynolds number hydrodynamics. This is the squirmer model. This is an organism. We conceptualize it as a spherical organism that propels itself with a tangential velocity boundary condition. Okay, so it moves by squirming and moving its boundary like a conveyor belt. And this might seem like a bit of a drastic assumption, but there are a whole range of organisms for which this model actually matches really well with the flow that they generate. And you can tune this system between a puller and a pusher depending on the sign of beta. That is the second term here governing the stresslet. In this system, we're going to be studying a monolayer, so a two-dimensional array, but in three-dimensional suspension of squirmers. Okay, so they're three-dimensional spheres confined to lie in a monolayer. And there are some interesting non-dimensional parameters here. For example, the swimming speed of the organisms non-dimensionalized by how we shear this suspension here with the shear rate. And we have an areal fraction phi. It's also possible in this model framework to actually subject these organisms to an external torque. Many of them are actually gravitactic. They can respond to the direction of gravity and swim along them, the field lines there. So we can actually have a gravitational direction here associated with this as well. And there's another non-dimensional parameter that measures how strong gravity is with respect to the viscous torques on these organisms. So as an umbrella, I wanted to show you basically two very distinct, but as it happens, quite similar methods for calculating the viscosity of the suspension. So we're only interested in the rheology at this point. So one thing you can do is go through this very well-established process of Stochastian dynamics. This is where you calculate the hydrodynamic coupling between all these spheres. So as they swim, they generate a disturbance around them that affects all of the other spheres, and so you need to solve it holistically all together. This is very computationally expensive, but very accurate. this method of Stokeskian dynamics goes back to work from John Brady and others, and has been extended to active particles very successfully. And the way you calculate the viscosity of this suspension then is by looking at the particle stress tensor after you've solved this. Now this is one way, but if we think about microscale measurements of viscosity, our mind often turns to a rheometer, okay, and the movement between two plates. And so another way that we'd like to conceptualize this is to use lubrication theory here. This is where we take a suspension of organisms, these active particles that are interacting hydrodynamically, but we only consider their interactions to leading order based on pairwise near-field interactions here, as I've identified by these red dots. Okay, and so in this case, we would subject this to a Couette flow, the movement of these plates. We calculate the hydrodynamic interactions using lubrication forces, which dominate in the near field, and then we measure the viscosity as you would in an experiment by calculating the drag on the two plates and seeing how hard is it to shear those plates, and from that we can back out the viscosity. So this is a very, very different conceptual frameworks for measuring the viscosity of the suspension. Just briefly, in the latter case with the lubrication theory, the way we do this is by propelling the spheres along with a driving force. These are subjected to a background force from the fluid, a viscous torque due to the vorticity in the flow that will tend to turn the organisms due to the shear. They're subjected to a gravitational torque, and this all goes into a resistance formulation. Low Reynolds number hydrodynamics tells us that they must be force and torque free overall, so their swimming speeds must balance all of these to give rise to a zero net force and torque. So if you go through all of this trouble, you can eventually, in these calculations, work out what the force on the top and bottom plates are, and therefore effectively measure the viscosity of this suspension. Now I won't go into all of the parameter variations we've tried, but I wanted to just really highlight the effectiveness of this approach. You can see on the left here, Stokeskian dynamics simulation from my collaborator Takuji Ishikawa, and then the lubrication theory simulation, which only takes nearest neighbor pairwise interactions into account, okay? Where the spacing between, you only count the forces when the spacing is less than 1 radii between the spheres. And collectively you can see very strikingly similar results here. But the question we wanted to ask is, how do these dynamics and the rheology of this suspension actually depend on things like the areal fraction, okay, so how densely you pack these, what is the activity of the swimmers as they start to swim faster, and other things like bottom heaviness. As we try and align these organisms due to an external torque and initiate some kind of pneumatic ordering, will that affect the viscosity as well? So this is a really interesting time-dependent problem, because the viscosity is not just a constant value. As these spheres in the highly packed regime actually slide and move past one another and periodically jam, you see these strong fluctuations in the instantaneous viscosity between the suspension. And you'll see in particular a significant difference between the case of inert spheres, where we switch off the swimming motion, and the case where the swimming motion is turned on, okay? So they're actually squirming. In this situation, you'll see, therefore, that the activity of these particles actually enhances the viscosity of the suspension. So it makes it harder to shear this active suspension. And one of the key parameters we would like to vary, of course, is then the areal fraction of spheres. So as you pack more and more spheres in, the viscosity goes up, and it goes up more for these particular parameters for the case of active swimmers compared to the passive spheres. Okay, but what's important, the more important takeaway message from this is that both the lubrication theory and the Stokeskian dynamics simulations, which are wildly different in the time it takes to compute them, give very similar results. So we've actually extended this to other kinds of problems here. We have a 3D simulation now of suspension of micro swimmers through a pipe. Okay, so this is a Poisson flow, and in this case, some perhaps peculiar behavior actually starts to emerge. And so in particular, what we can see is that the viscosity in this situation can actually go negative. So here you have an interesting competition for flow moving upwards. The viscous torques can align the cells to swim away from the center of the channel, balancing gravity, and this can actually then help to propel that suspension through the channel, okay? Because as they're swimming, they're pushing on the walls and helping the fluid to move through the channel. And so this just points that there are some really subtle but very interesting physics going on here, I guess, in these suspensions. And actually, this can have a negative viscosity that actually tries to flow more easily than the solvent viscosity, actually providing energy, if you like. So that's all well and good, and we can do pretty well in measuring the actual bulk properties and the rheology of this suspension. But what about the actual dynamics of individual and pairs of cells? And so this is a project that a student in my group a few years ago, Chris Darvenica, embarked upon, and he looked to see whether if you have two of these individual swimmers moving along, whether their scattering dynamics and their pairwise interactions could be captured as well by lubrication theory rather than resolving the full hydrodynamics. So we'll compare these results here where we switch on interaction at one sphere radius with cases for full boundary element simulations. So these were conducted by Takuji Ishikawa and go back to this seminal work that they did almost 2 decades ago. So what I'm showing here are a series of videos for different parameters where we have different values of the squirming parameter beta, and we're the switching on or off the effect of gravity in the background as well. And you can see some really interesting dynamics from this sort of pairwise trapping organisms that swim along as a pair in a bound state forever. Perhaps they scatter in some very in a non-trivial sense here to some interesting outgoing angles. Perhaps they orbit one another indefinitely like this, or in the case where you switch on gravity, this orbiting can exhibit a drift upwards, okay? So gravity is trying to move them upwards. And so if we put all this together, it's possible to go through and really characterize the outgoing angles as a function of the incoming angles of these micro swimmers. Okay, so we've seen some really interesting talks already in this conference about how particles that are interacting through various potentials as a function of distance only interact. In this problem, it becomes a lot more complicated because now we have an angular dependence on these interactions. But nevertheless, it seems to capture the dynamics really quite well. So I'll compare here, just for completeness, what we see if we compare the lubrication trajectories with those of the boundary element simulations. These are pairwise comparisons between the two papers, and you can see that the scattering is, again, captured pretty well. Not across all parameter regimes, but many situations, the lubrication is enough to actually capture this. And so this model here of the interacting microswimmers will be useful for the next phase of the talk. But what I'm going to do, first of all, is introduce this organism, okay, and the background. So we have this organism, magnetotactic prokaryotes, okay? Now these are a class of microswimmers that actually create and they build a magnetic compass needle within their cell body. And they do this. This has been well understood for unicellular organisms for a few decades now. So there's some beautiful experiments in the literature about unicellular magnetotactic cells, like the one shown schematically here, that they fabricate this magnetite inside the cell, and this helps to align them passively through the magnetic torque along a field line. Then they just swim along that direction. Okay, so they have a flagellum on the back that propels them, and through just physics, okay, undergraduate physics, they align with that field and they swim along that. And they use that to burrow down into the soils and sediments where the oxygen conditions are favorable for their growth. But there's actually a class that's discovered fairly recently where these organisms are actually multicellular in an obligate sense. So here's a colony that is composed of sort of 5 to 20 of individual cells bound in a spherical state. Each one of these cells builds the compass needle inside it, and collectively it therefore has a magnetic dipole. Okay, shown in this or a magnetic moment. Sorry, shown in this red direction here. Now this organism is covered in little cilia, flagella, that help to push it along, and this coincides with the direction of the magnetic moment. But there's some really interesting questions about how these behave and how a multicellular colony like this can actually navigate the magnetic field lines and what other conditions it might respond to. So I'm going to show you some work now. This is work led by a PhD student in my group, Xinyi Yang, and in collaboration with Manu Prakash at Stanford University. And what we did was to go and actually look at samples in the wild and identify these multicellular magnetotactic prokaryotes, or MMPs, as you might come across them in the literature. And we put these in a droplet of water, okay, under the microscope, and subjected them to a magnetic field. Now, if you just think about the physics problem I outlined before, you would expect that these would just accumulate at the boundary here. Okay, so you've got water, you've got an air interface, and you would expect them to just swim to the left and just sort of push against the boundary there and stay there indefinitely. Okay, but what we saw was something really quite interesting in that these jump back and forth in this periodic sort of popcorn-like manner. Okay, so they don't accumulate at the boundary at the air interface where the magnetic field would guide them alone, but they periodically jump back and forth in this kind of ping-pong locomotion. And so what's actually happening here is that the magnetic field is guiding these active particles, these bacteria, to the left, but there's an oxygen gradient that is pointing in the opposite direction. And this is a very confusing situation for these bacteria that usually use the magnetic field to guide to favourable oxygen conditions, but in this case they've been tricked because when they get to the interface there's actually a higher oxygen. And they're able to temporarily override their guidance due to the physics of the magnetic field and swim in the opposite direction. Okay? But they have very short memory, and so eventually they move away and then they come back. So what we did was to go through and analyze the trajectories across many cells, many colonies in these videos, and we can see, for example, that an organism that's jumping out, okay, there's escape mode, there's jump, and then it waits for a bit and then it starts to come back, okay, as it forgets that it was a terrible idea to go to the oxygen interface. And we trawled through many trajectories here and found that, probability distributions for the distance that these organisms jump. Okay, this is D jump, how far it comes out, and the waiting time that it exhibits at the boundary before it jumps, so how long it takes for it to realize that the oxygen conditions are very bad and move in the opposite direction. And then we compared... Yeah. These organisms prefer low oxygen environments, but we have presented them with a high oxygen stimulus, which is opposite to what they would expect, I guess. So they're guided by the magnetic field, and then because of the oxygen diffusing into the water there, provides a stimulus for their escape mode. Yeah.
So the experiment was almost to be, because sometimes it seems to disappear, so they go out.
Yeah, it's a good point. So this is in a hanging drop kind of experiment where you have a sort of tapered edge there. So we've confined them to lie in a pretty narrow plane there.
Because in the movie sometimes they disappear.
Yeah, they move slightly out of plane. That's okay. Yeah, yeah, exactly. So we compared the observations with an agent-based model where we treat these organisms initially as point-like particles. Okay, so they just have a position X and Y. They have some. We ignore the 3D effects here for their orientation, but they have some angle phi with respect to the magnetic field. So one of the key parameters here, K, is the relaxation timescale, basically, or of the microswimmer, basically balance between the viscous torques and the magnetic torques. Okay, and we put in a simplified model for oxygen sensing. Okay, so they can basically stochastically change direction based on whether or not they presented with favourable conditions, or they detect that the oxygen concentration is favourable or not. And this, we can simulate trajectories of particles in this model framework, and the simulations agree pretty well with the experiments, as you can see here, just from the trajectories alone. But we can do a bit better and go through and actually compare these probability distributions. So what I've shown here is the result between the experiments in blue, and then simulations for, we've got 150 of the bacteria, and then 15,000 with the black curve. Okay, so it matches pretty well what we see for both the D jump, the distance that they jump, and also the waiting time before jumping there as well. This large peak here that might interest you is actually because of the finiteness of the simulations, and so these are just cells that don't jump in that window. So you need not be worried about this large peak at the high value of t. But all the other parameters we've extracted from the experiments and put into the model here. Things like the movement, the diffusivity of oxygen, the profiles, and so on. So that's all well and good, but you might ask what happens at the interface where these cells really crowd up and form what looks like a pretty dense monolayer. And so presumably a point-wise accumulation of particles is no longer valid in that regime, where you would expect actual interactions between cells in this densely packed environment. And so what we did then was to actually use the models I was outlining before with these hydrodynamic interactions, these lubrication forces, and actually implement that into this system here, where these organisms are now also subjected to a magnetic torque that will try and align them upwards in this simulation. These squirmers here are then color-coded based on their orientation, okay, so just with a periodic color map based on their angle phi. So what you'll see is that overall, for quite a weak magnetic field, the organisms are guided upwards by the magnetic field. They tend to accumulate a little bit, but they're also dispersed through interaction. So when they crash into each other, the hydrodynamic interactions can destabilize their orientations and deflect them and push them away such that they move away from the boundary, as you can see here. But following this cell here, you'll see that it actually turns around and eventually makes its way back. So there's sort of a loosely dispersed state here, which is guided gently by the magnetic field to push them upwards. If we go in the extreme opposite direction here and turn on the magnetic field very strongly in these simulations, then unsurprisingly, they're very much confined to lie in the same direction as the magnetic field, and so they all swim upwards and they pack here, all pointing upwards. Okay, so these are the two kind of extreme cases, I guess you could say. You might see, if you look really closely, some small fluctuations here in the orientation. Okay, so it's not all the same color. So there are some.
Why there is overall drift towards right?
I beg your pardon?
Why there is overall drift of the particles towards right? Because it's
Yeah, okay. Well, this has periodic boundary conditions, the simulation on the left. So there's going to be, on average, there'll be no drift left or right, but this would be just to the stochastic realization of this particular simulation. Bear in mind, in this case, the magnetic field is upwards, not to the left. So the drift you would see would be up, and there's equal probability of them being a small bias right or left based on the sample size here. Okay, so there's nothing special about that realization. Okay, so these are the two extreme cases, but what you might ask is what happens now as we vary this magnetic field strength. Okay, so really this is just a non-dimensional parameter that, as I said, compares the viscous torques through the hydrodynamic interactions with the magnetic field strength. Okay, and what you can see here, I've turned them sideways again now, so the field is going to the left. In this case, for very weak magnetic field strength, they loosely accumulate in this kind of dispersed manner near the boundary. And then as you increase the field, you guide them more and more to the left here and result in this strong packing ultimately here. Okay, so it was pleasing now to go back and compare the probability distributions of these microswimmers here with the experimentally observed ones and the point model. So you can see here there's actually a pretty good agreement between this model here with the full hydrodynamic interactions between the organisms and the case where we just treated them like point particles. But there are small differences, of course, based on the fact that these organisms can't, when the hydrodynamics is included, they can't move completely independently of one another. Okay, so cells that are at the interface and trying to escape have to make their way through a dense lattice to do so. So there can be some small perturbations there. But perhaps what is more interesting is when we looked at the order parameter here. Okay, and so this really tells us something about the collective alignment of organisms. So an order parameter of 0 tells you that they're isotropically distributed, randomly aligned. An order parameter of 1 tells you that they all have the same orientation. And unsurprisingly, as you increase the value of the field strength, shown here by K, this order parameter in black increases monotonically on average, so that's fine. We expect that. But what's interesting is that if you look at the standard deviation of the fluctuations here, so the time-dependent order parameter, this has very strong non-monotonic dependence. So these will be very small in the case of low field strength because they can move around independently. When the field is very, very strong, it suppresses all fluctuations, so it goes back to 0. But there's an intermediate case where the field is strong enough to guide them and align them near this interface, this oxygen interface, but still not strong enough to completely overcome hydrodynamics. Okay, so it brings them together in a manner that can elicit hydrodynamic interactions, which then can destabilize their orientations. And so you see very strong fluctuations in their alignment, their ordering in this particular case. So I'm going to move now onto the final sort of chapter that we've been looking at, and this is to really investigate how organisms can navigate chemical microenvironments. Okay, so whether it's in the ocean or groundwater, all sorts of aquatic systems, medical devices, bacteria are confronted with microscale chemical gradients and small hotspots. And their ability to swim towards these food blobs, maybe they're just a few tens of micrometers away, can have a tremendous effect on the nutrients and the food that they can acquire, and therefore their growth. So this process has been pretty well understood, this process of bacterial chemotaxis. You subject them to a gradient, they move up this gradient, and that's all great. But most of the work that's been done in this space has looked at organisms which are responding to very strong chemical gradients. Okay, so and this has been done, for example, very strong hotspots here or around large organisms exuding lots of nutrients. And there are many situations where this is the case. Here's a large diatom that's leaking nutrients into the environment. You can see little bacteria swarming around this and feeding on the nutrients that are leaking out of this cell. Okay, so that's a very strong response that you can see. But there, many of the organisms that you would see in the ocean, for example, would be really microscopic targets. And in fact, the bulk of the biomass in the ocean really is composed of targets that are the same size as these bacteria. So the question we wanted to ask was, could bacteria use the chemotactic response to navigate towards really microscale hotspots, about the same size as themselves? And so we developed, this is work in collaboration with long-term collaborators at ETH Zurich, and experiments led by Francesco Carrara. And so we developed this microfluidic platform that enables us to actually place in or release a microscopic quantity of nutrients about 1 million times smaller than a single grain of sugar. Okay? And we did this not by dropping in a blob of food, but by using light to actually release nutrients. Okay, so a small pulse of LED light, as you can see in the video on the bottom left here, will create a microscopic blob of food less than a picomole. Okay, pico is not a SI prefix we see very often, but less than a picomole of nutrients here to represent the same quantity released by phytoplankton. And then we watched using a video microscopy how the bacteria would respond to this. Something that's really important at this scale is that the idea of chemotaxis, okay, or responding to a chemical gradient, actually becomes a little bit more difficult because cells do not see a smooth differentiable gradient. This is a very stochastic process. Okay, so actually the individual molecular encounters associated with this gradient become important, and fluctuations in that give rise to actual uncertainty in that gradient estimate. Okay, so it's a stochastic process. And we've incorporated that fundamental limit to gradient sensing into a mathematical model. I won't go through all the details, but it's in the paper down the bottom here. I'm happy to chat later. But the long story short is that this model can recreate the observations from our microfluidic device to less than 1% fitting error. Okay, so we have a really good understanding of how these active particles can respond to a diffusing chemical hotspot. As shown in this video here. Now that's all well and good, but you might say, well, in nature, surely a hotspot is not just a solitary individual blob of food, but rather a more complicated, structured environment. And so over the last few years, we've been working to extend this modeling framework and the experimental framework to produce a long-term statistically steady state environment composed of multiple ephemeral patches. And so this can be done for many hours now. So you can release more and more blobs, hundreds of them if you like, over a long period of time, and watch how bacteria, these active particles, actually forage in this environment, going from hotspot to hotspot. Okay, so the beautiful thing about this is that we can connect the individual behavior of microswimmers at the scale of milliseconds and seconds all the way through to their long-term dynamics and growth properties and other things, really bridging those ecological timescales. And this has really not been done before in a manner that can have a dynamic, unsteady environment. Usually the unsteady environment is very short-lived. You can watch it very quickly and then it's over. So here it's both unsteady, dynamic, but also over a prolonged period of time. And so we can now think about modulating the spatial structure of this environment and releasing things at predetermined locations, changing the pattern and the statistics of this foraging environment. I won't go into all the details, but it's basically a very rich playground where we can collect trajectories for millions of cells as they're foraging in this structured landscape. So, I beg your pardon, sorry?
So can you also monitor the concentration of the nutrient?
Yes, yeah, absolutely. So we can monitor the concentration of the nutrient. We do so by, we know how much we inject at different points, and so we can reconstruct the whole chemical landscape by solving the diffusion equation effectively, and we've calibrated that really carefully. It works very well. So the really powerful product of this setup really is that we have the entire chemical and resource landscape at all points in time, and we know where all the cells are at all points in time. So we can look at partitioning resources among the population, how they respond to different gradient stimuli, and really overlay the resource landscape and the positions of the cells. Yeah. Yes, we can, yeah. So you're right that they do consume the nutrients, and that's possible to affect this. In this case, we have a bacterial concentration which is sufficiently low that it doesn't really perturb the underlying nutrient structure. But we've looked at problems where that's included as well. Okay, so again, you say this is another step towards reality by including a spatially structured environment. But what about real target cells? Because they don't all respond to glutamate that's released in the lab, but rather through the nutrients being exuded from individual cells. And this is particularly important because up until just a couple of years ago, people thought that the organisms really of this size were the sort of limits to where bacteria could detect chemotactically, even though the bulk of the biomass in the ocean really is composed of these really small microscopic cells, things like Prochlorococcus and Synechococcus, okay, just a couple of microns. So there's been a sort of perceived boundary here for the detection limits. But owing to what we've just seen on the previous few slides about how accurate these cells are and precise they are in detecting gradients, we wanted to see whether they could find these really small cells. And there are fundamental differences between detecting large chemical hotspots and small ones. If you have a large hotspot and you're swimming around, you experience this nutrient-rich exuding environment really as a pretty smooth gradient of which you can differentiate. If you look at sort of a time series of encounters, you see this sort of gradual increase in molecular encounters. And this is really different if you have a small target, okay, where the sort of random walk that the bacterium will execute to find this target has a mean free path which is commensurate in size with the target you're looking for, okay? And so this causes really like no signature for a while and then all of a sudden a bit of a blip. So there's a really pushing the boundaries of where you could detect here. So armed with this sort of hypothesis that cells might be able to detect really small targets, we set about doing some experimental work and modeling, and this is work with Jean-Baptiste Reynard in Justin Seymour's group in UTS. And we actually took the bacteria that can swim around and nutrient targets here shown with Synechococcus, and these cells are labelled with isotopes of different compounds. So these are enriched with carbon-13, well above natural abundance. These cells, the Synechococcus, were enriched with nitrogen-15, well above natural abundance. And after that, we took these cells, we grew them up, we washed them, and we put them together for 3 hours. And then afterwards, we looked at how much of these isotopes were present in the other cells. So the idea being that if there's any enrichment of N15, above natural abundance in the Marinobacter, it would have had to have found them from the Synechococcus. Okay, so that's basically like a follow the money sort of approach of actually tracking the fate of nutrients going from one cell to another and attributing that to the spatial encounters between these organisms. But the key thing that we were able to vary here is switching on and off their capabilities to do chemotaxis. So we have bacterial mutants that can do chemotaxis, shown on the top here. We have bacteria that cannot do chemotaxis, so they do an unbiased random walk in their movement, and then cells that can't swim at all. And what we saw now is that the amount of nutrients absorbed by the bacteria foraging in this environment depends, first of all, very strongly on how many target cells there are, and that's not surprising. The more of the hotspots you include, the more nutrients available to the cells looking for them. Okay, so that makes sense. But what was really striking was that if we looked, that was for the cells that can do chemotaxis, if you switch off the chemotactic capability, or you switch off their swimming motion altogether, you see a significant drop in the amount of resources that they can acquire. Okay, so this is really the first evidence, direct proof that chemotactic bacteria can actually find more nutrients than non-chemotactic counterparts, even for targets that are so small that they're about the same size as an individual bacterium. So this was a very exciting result. We then also showed discovered that there was a reciprocal nutrient exchange, and that in finding these blobs of food, the bacteria were also providing nutrients to the Synechococcus cells. So this is a two-way exchange of nutrients mediated by this highly ephemeral encounters between cells. I'll just briefly outline the mathematical modelling that we used. Effectively, we're able to model this very accurately with conceptualizing it as a cell that's leaking nutrients spherically, symmetrically into the environment, and it produces a radially symmetric profile of concentration of nutrients. Here, I've shown a two-dimensional slice through this 3D landscape. So this is the environment in which the cells are foraging. And I compare here, just for demonstration purposes, what it looks like when you have chemotactic bacteria on the left, looking for phytoplankton cells. Non-chemotactic on the left, which is searching for phytoplankton. Okay, not much happens. Okay, it's just isotropic swimming. But if you switch on the chemotaxis, you can see that this clustering occurs around the hotspots. Okay, so they're able to hone in on their target cells. In this case, I've accentuated their abilities for demonstration purposes, okay, just to show you what this kind of clustering looks like. But if you fit the model to experimental parameters, then the chemotactic case actually looks more like this. Okay, so you can see this is the case with chemotaxis. There are interactions here, but if you just look at the video, it doesn't look like much is happening. Okay, you'd be hard pressed to see any kind of accumulation or net drift of these particles towards these basins of attraction in blue here. But nevertheless, from a statistical point of view, the encounters are there. Okay, so if you look at the actual resources that these cells acquire, and you average this over time, you can still see a strong signal in the uptake compared to the non-chemotactic cells. So this is a very weak effect, and it's really sort of pushing the boundaries of when chemotaxis is possible. The last thing to just go through really quickly is that in our model, we're able to investigate some of these spatiotemporal interactions between these cells. Okay, so we have a target cell here, the bacterium swimming. We can count how long it spends in the vicinity of its target, the residence time, and then look at a probability distribution of those interaction times. So what you'll see is that going from the non-chemotactic in blue to the chemotactic one in green actually increases the average residence time. So those that are searching for food particles can actually spend a little bit longer there, okay? 3 seconds instead of 1 second. But this is still relatively short-lived, okay? So there's still a fleeting interaction going on here. It's not a permanent kind of trapping. You found your food and you're attached to it forever, but rather a transient interaction. That's for each of the interactions, but we can also go a bit further and look at how many of these target cells a bacterium will find over the course of a day as well. And so by switching on chemotaxis, you promote not just longer encounters between these particles, but more encounters overall as well. And so this actually points to a really interesting conclusion that cells are able to exchange nutrients in a kind of form of symbiosis, but in a manner that doesn't require them to kind of stick together and form a permanent association. They're able to transiently swim past one another and have the passage of nutrients through these spatial interactions, even though they're very short-lived. So really just a couple of quick messages that I wanted to leave you with today. First of all, that in these intrinsically non-equilibrium systems of cell motility, we can further perturb these systems by investigating the role of moving boundaries and fluid flows, competing fields, be it magnetic fields or oxygen fields that might compete or conflict with other signals that they receive, and creating custom chemical landscapes that start to probe how organisms can navigate realistic environments. So I've put a few key papers from this work up here. I'd really like to thank my research group at the University of Melbourne, funding sources, and many people who've contributed to this work. In particular, Chris Darvanisa, Xinyi Yang, and Francesca Carrara in the multi-patch problem there, as well as a whole range of other co-authors. So thank you very much for your attention.
I had one question. So in general, in general, one imagines that there is a food chain, but here the people are exchanging food. There is no hierarchy. How common is this generally in biology?
That's a very good question. Yeah, so when we think of the food chain, we often think about this as a hierarchical process, that something larger will kind of eat something smaller, and we have this kind of trophic levels, I guess, of different layers there. So really, I guess what we're discovering is that in many of these situations, for example, like a microbiome or in a microbial community, you actually have a significant array of exchanges and interactions between organisms of the same size. And that's particularly true at this microscopic landscape, where cells are sort of competing for resources, they're sharing chemicals. The waste product of one might be a growth promoter for another. And so they're very strong interdependencies in this food web, even at the same kind of level, if you like. Yeah.
Okay, thank you. Yeah, sorry.
So you mentioned that in the ocean, like these food sources are like sizes are very small, like the same size as the bacteria or something. So is chemotaxis the only thing, or also there are some other mechanism by which because it seems like you said the chemotaxis is also very weak, so it seems like almost like, you would not hit a food source at all.
Yeah.
Right? So is there also any other mechanism apart from chemotaxis? Or?
Yeah, so chemotaxis can be quite weak at this scale, but it still has a payoff ultimately, right? So you saw a significant, in some cases, a significant enhancement in the resources that they acquire due to chemotaxis. This can obviously depend on the abundance of nutrients. If there's nothing around, then technically it's not worth your effort doing chemotaxis or even swimming. And so there are many cells that don't bother swimming at all, and they just exhibit a minimalist kind of lifestyle, incorporating nutrients from the environment. So there are all sorts of different sort of game theory strategies, I guess, for uptake of nutrients. And we've looked actually at some of the payoffs with respect to the cost as well. I didn't talk about it today, but you can implement the cost of swimming energetically.
In the bacteria where those which respond to magnetic field, and have you looked at if the magnetic field change, for example, periodically or something, so they can make rotations or something?
That's a good question. Yeah, the videos I showed today, we have a field which is permanently there, permanently switched on, but we have done experiments where you can remove that field and then you see the kind of breakup and dispersal of that cluster. So using things like permalloys and other systems, I guess, you can actually create a really interesting range of transient magnetic fields and sort of periodic switching and control them. It's not something that we've really looked at, but I think it's a really interesting area to think about how faithfully they can follow, for example, dynamic fields. Switching, yeah.
So, if I got it correctly, so in your case, the nutrients were always provided by the environment. But can you also consider the case in which the particle itself is releasing nutrients which is felt by the others?
Yes, so
solve chemotaxis if you want.
yeah. So in the last case, we were showing nutrients that were being released by one particular chemical, one particular species, and then absorbed by another. So they were actually released.
So the simulations where you show this aggregation were in that case.
In the experimental platform and the simulations where we had the multiple hotspots in space, these were released in a laboratory setting. Yeah, but they're designed to mimic the release by individual cells. And so you can either do that as a all-in-one, like a spontaneous burst or release, or you can have a sustained release. But in that case, they're designed to mimic how a cell would be releasing nutrients into the wild in a steady state manner.
Okay, thank you.
Just a question in line what Sanjib asked. Some years ago, Vergasole and Schreiman studied the problem. The motivation was a little bit different. You look on the bats and how the males feel the females. And so there are pheromones, there are plumes, and the medium, of course, subject to some kind of turbulence and so on. So the information they receive are tiny bits and they're incredibly sparse. And they came with a model which they called infotaxis. And I'm curious if it can be adapted precisely to your situation when you lack chemical gradients.
Yeah, that's a very good question. In these environments, we're operating in a regime which is below what's called the Kolmogorov length scale. It's where the velocity, the fluid velocity tends to disperse chemical gradients into these plumes and trails and all sorts of interesting kind of turbulent structures that you were referring to. At the scale beneath that, sub sort of millimeter scale, things actually are much more well-behaved with respect to diffusion. And so the environments that the chemical and microenvironments that these cells experience are much more sort of laminar flow, gentle shear, and more sort of regular diffusion. The other problem that means that you can't integrate over long timescales is that these organisms are fundamentally constrained to their sensory timescale of sort of fractions of a second. They don't have a long memory, so they can't sort of count information over long periods and see a really much more elaborate plume trail.
Integrate and go. Yes.
Yeah, so you can really only integrate information over a time scale which is commensurate or shorter than your the time associated with your random walk, right? Okay. Otherwise, you're just conflating information. So you need to be moving ballistically while you are integrating information to make sense of that. Otherwise, if you change direction and you're still integrating, then you effectively get like a motion blur. You can't make sense of that gradient. So, but it's interesting. I think there are some interesting links.
Yeah. So I want to ask, you told about two types of simulation approaches. So one is the usual way, taking all particle interactions. And the other one is the the lubrication one. So in that, I did not understand. So when you had shown for the usual case, you take the shear flow kind of thing. And in the lubrication one, you take some other flow. So some covert flow or something.
Yeah.
Why is it so? I mean, can we not take that shear flow itself in the lubrication theory?
So that's a good question. So in each of these cases, you can study whether they're the Stochastic dynamics or the lubrication theory. You can study the behavior of ensembles of these organisms in a quiescent flow where there's no external flow, or you can impose whatever you like, basically. So you can have what's called a couette flow, which is just a shear flow, or you can have a parabolic flow through channels. You can impose all sorts. And so we did that for both cases. There's no difference in the imposed flows that we use.
Okay, so the answer was independent of the imposed flow, was it? I mean.
Yeah, so the way that we measured the viscosity was in this kind of linear shear flow. But there are in complex systems like this, there are multiple ways to get at the viscosity or the rheology, right? In the simplest case, we've studied what happens when you shear the system. So that gives you a shear viscosity. And we compared it like for like between those two.
Okay, one more. So in the bound states which you find, so there how do you confirm their bound states? So you run for a long time or some...
Yeah, we run them.
Or you plot some probability distribution or something?
Yes, so in those hydrodynamic bound states, and I should mention that these organisms are large enough, typically, that you can neglect thermal fluctuations in terms of rotational diffusion. In that situation, we basically looked for convergence of their relative orientations and identified them as bound states. Effectively fixed points in a dynamical system of their orientations.
So you considered them as point particles, or you took the volume exclusion?
No, volumetric exclusion, yeah. But they each have a position and orientation. So a bound state is when their relative positions don't change. So you just have like solid body rotation.
No. So,
yeah.
Okay, fine.
Yeah. Happy to keep chatting though a little later. Thank you. Okay.
Okay, let me begin by thanking Sanjib for giving me this opportunity to present some of the work this ongoing work, part of it, to this nice meeting, and look forward to feedbacks from you on this work. So this is a work which is on collective motion of multi-motion and transitions in a collective